My name is Jack Szostak. I'm a professor of genetics at Harvard Medical School and an investigator at Massachusetts General Hospital, where my laboratories are. I'm also an investigator of the Howard Hughes Medical Institute. And in this lecture, I would like to tell you about some of our recent work relevant to the origin of life on Earth. And we're going to focus this time on the membrane component of simple protocells, how those membranes assemble, what their properties are, how those properties are relevant for early forms of life, and how we imagine them gradually changing as a result of the emergence of Darwinian evolutionary processes. So let's begin by having a look at our conceptual model of what an early protocell looks like. So again, we think of protocells having two parts, a membrane boundary that closes up into a spherical vesicle that can encapsulate molecules in the internal aqueous compartment. And the most important of those would be genetic molecules, possibly RNA or DNA or maybe some related kind of nucleic acid. And what we would like to understand is how, in the absence of highly evolved genetic material, this membrane compartment could first grow and subsequently divide into daughter cells. Okay, and because we're thinking about this at a very early time, prior to the emergence of Darwinian evolution and advanced, highly evolved cellular machinery, we have to look to simple chemical and physical processes to drive these transformations. Okay, so to think about that, we need to think about the molecules that are going to be used to build these membranes. And our favorite molecules in this regard are the fatty acids. So these range from short-chain saturated fatty acids like capric acid, which are the kinds of molecules that we think could have been easily made in different scenarios on the early Earth. Many times in our laboratory work, we use more modern fatty acids, myristoleic acid or oleic acid, simply for convenience. But in the end, we always come back to more prebiotically reasonable systems. So what are the biophysical properties of these closed-up membranes in the form of vesicles or liposomes? So the most important aspect, and something which is quite different from modern membranes that are based on phospholipids, is that fatty acid vesicles are basically, their formation and existence is controlled by a series of phase transitions. So, and the most important of those depends on the pH of the environment. So at fairly alkaline pHs, the carboxylates of fatty acids are basically fully ionized. And then charge repulsion wants to keep those groups as far apart from each other as possible, while the hydrophobic forces want to keep the hydrophobic tail of the molecule as close together as possible. And the result is the formation of micelles, which are very small aggregates, a few nanometers in size, and composed of roughly 10 to 100 fatty acid molecules. Now, as the pH in the environment gradually drops towards roughly 8.5, which is the point at which half of these carboxylates will be ionized and half protonated, there is a phase transition that results in the spontaneous assembly of bilayer membranes, as you can see here. And because the curvature is much reduced in these much larger aggregates, these are actually stabilized by hydrogen bonding interactions between adjacent charged and ionized carboxylates. Now, as the pH drops even further into the acidic range, all of the carboxylates will become protonated eventually, and then these structures collapse into oil droplets. So the existence of these bilayer membranes is only possible at an intermediate pH range, close to the pKa. You'll notice this pKa of 8.5 is considerably higher than the pKa of individual fatty acid molecules in solution, which is closer to 4.5. And the reason for that is that these charged head groups are much closer together in this aggregate structure. Okay. Now, when you do form these membranes, they have very interesting properties, which again are very different from the properties of phospholipid-based membranes. And one of the things that is very important to us is the dynamic exchange processes that occur in these membranes. So fatty acid molecules in a fatty acid membrane flip-flop back and forth from the outer leaflet to the inner leaflet very rapidly, sub-second time scale. They also exchange in and out of the membrane, so into solution where they can exchange with molecules in micelles. And again, this is a very rapid process so that the molecules that make up any given vesicle are constantly in flux and exchanging with the molecules in other vesicles, again on the second time scale. Okay, another very interesting aspect of these membranes is that they can be quite heterogeneous at a molecular level. And so these membranes can incorporate not just fatty acids, but also their glycerol esters, as you see here, and even lysophosphatidic acids, so ester with phosphoglycerate. They can incorporate alkanes and polycyclic aromatic hydrocarbons as well. Now, what happens when you make membranes with these kinds of mixed compositions? It's very interesting that in many cases, this results in membranes that are much more stable. They're more physically stable, more thermostable, and also often more permeable to polar nutrients that we would like to bring in from the outside environment. And so this slide simply illustrates an experiment to look at the thermostability of some vesicles made from a pure fatty acid as opposed to mixtures. So in this experiment, what we've done is to encapsulate a short DNA oligonucleotide within vesicles and watch it leak out over the course of about an hour. And now, if the membrane is composed purely of myristoleic acid, this 14-carbon singly unsaturated fatty acid, you can see that up to about 50 degrees, nothing leaks out over an hour. But as the temperature goes up to about 80, they become more leaky, and then at 90 degrees, everything is leaked out over an hour. Now, if you add some of the corresponding alcohol, you can see significant stabilization. But the most dramatic effect is when you add a fraction of the glycerol ester of that fatty acid. And now you can essentially boil these vesicles for an hour, and none of the contents leak out. At least these oligonucleotide contents are trapped inside. So that's really exactly what we want. We want the genetic material, long oligonucleotides, to stay inside indefinitely, while small molecules, nutrients, can get across the membrane very rapidly. And again, as the temperature goes up, the permeability to small molecules increases dramatically. Okay. So we got into this area about 10 years ago and started doing some very simple experiments based on the pioneering work of people like Dave Deemer and Pierluigi Luisi. Now, the Luisi lab in particular had shown already that you can make vesicles grow simply by adding more fatty acids in the form of alkaline micelles. When the micelles go into a buffered solution, they become a thermodynamically unstable phase. So there's an energetic driving force for those molecules to assemble into either new membranes or to integrate into pre-existing membranes. And at the time, Marty Hanchik was a postdoc in the lab, Shelley Fujikawa was a student. They basically repeated those experiments but using real-time methods of analysis so that we could watch the process of vesicle growth in real time after the addition of food molecules. The assay that we used was a fluorescence assay based on energy transfer from donor to acceptor dyes. It's basically a measure of how far apart the dyes are on average. And as the membrane grows by incorporating new molecules, the dyes are diluted, they get further apart, and energy transfer efficiency decreases. So you can use this as a very effective assay for surface area in real time. So we can watch things grow very easily by this simple fluorescence assay. And the essential result from that is that if you add the food slowly, almost all of it gets incorporated into pre-existing membranes. If you add it very quickly, only a fraction, maybe half, will get incorporated, and the rest will form new vesicles. Okay, so that takes care of growth at a basic level. What about division? So in the early days, we didn't really have any good idea of how to drive protocell division through environmental fluctuations. So essentially, in desperation, we resorted to a brute force approach of forcing big vesicles through small pores in a filter, and little vesicles come out the other side. So on the one hand, it's satisfying because it's a proof of principle that cell division could be driven by a physical environmental force. On the other hand, it's not very satisfying because we didn't think there was any realistic analog to that process on the early Earth. And furthermore, when you take a large vesicle and physically divide it into smaller spherical vesicles, you necessarily lose some of the contents, which is also not very satisfying. Nonetheless, that process could be turned into a cycle where we could do growth and division and growth and division again and again. And so then the problem really became, how could we find other pathways that might have been more physically realistic, more robust, and more plausible for the early Earth? Okay, so the first advance in this area came from thinking about an alternative growth pathway. And so this is a pathway of growth that, instead of relying on the environmental influx of new molecules, occurs in a different way by competition between protocells in a population. So some are going to grow at the expense of others. And this was work done by Irene Chen when she was a student in my lab, and initiated by discussions with Rich Roberts when he was a postdoc in the lab. So the basic idea was that if you think of our basic protocell model, we have genetic molecules trapped inside a semi-permeable membrane. Now, large polymers contribute very little to the overall internal osmotic pressure in a system like this. Most of the osmotic pressure actually results from counterions that have to be there to neutralize the charge on the polyanionic genetic molecule. And so this is the classical Donin effect. These ions contribute to an internal osmotic pressure. And we thought that as a result of this physical phenomenon, this physical effect, vesicles that had more RNA inside them should have a higher osmotic pressure, and maybe that could actually drive growth competitively. So the way that we looked at that experimentally was using the same fluorescence assay that we had been using before to monitor membrane growth following the addition of new membrane molecules. But in this case, the idea is that we're going to take osmotically swollen vesicles that have, for example, a lot of RNA inside, and we'll monitor the surface area of those vesicles following mixing with vesicles that are osmotically relaxed. And we'll ask what happens to the surface area. Okay. So here's the experiment where we're monitoring the surface area of the swollen vesicles. These are vesicles that have a lot of RNA on the inside. And by themselves, they're perfectly stable. Nothing happens. Same thing for the relaxed vesicles. Nothing happens. As soon as you mix the swollen vesicles with relaxed vesicles, you can see that their surface area starts to increase over a period of minutes. And in control experiments where we mix with buffer or other swollen vesicles, there's very little change. Okay, so it seems like the swollen vesicles are growing by absorbing molecules from their relaxed neighbors. We can, of course, do the converse experiment and monitor the surface area of the relaxed vesicles. And when they are mixed with swollen vesicles, their surface area declines. So molecules are leaving those vesicles and going into the swollen vesicles. Again, control experiments where relaxed vesicles are mixed with more relaxed vesicles, not much happens. Okay, so what's going on here is that as soon as we mix these populations of vesicles, the swollen ones are growing, the relaxed ones are shrinking. And the reason that's happening is that this osmotic pressure creates a tension in the membrane, which is only relaxed by the incorporation of more molecules, which allows the surface area to increase. So the exciting implication of that physical process is that it's a potential link between genome replication and faster membrane growth. So any process that results in faster replication of the genetic material inside a vesicle will automatically, as a result of the Donin effect, lead to faster growth of the membrane compartment. So it's a coupling between genome replication and cell growth as a whole. Now, that could be particularly interesting if genome replication is an autocatalytic process driven by RNA-catalyzed RNA replication. So now, any mutation that leads to faster, more efficient replication of the genetic material will automatically go along with faster growth of the cell as a whole, and it's a competitive form of growth that results from these cells essentially eating their neighbors. Okay, so that was a very exciting conceptual advance, but there was always this nagging problem that here we're looking at growth driven by osmotic forces, which means that these are growing as spherical vesicles. And dividing a spherical vesicle, essentially one that's osmotically swollen, is something that's actually quite difficult to do. It takes a lot of energy, and as I said in the case of the extrusion experiments, results in loss of some of the contents, which is not very satisfying. So we were left thinking for a long time about possible alternative membranes and that alternative division processes, and that's something we'll come back to later. Okay, so let's think about more natural systems. Everything that we've looked at up till now has been a highly constrained, a very artificial laboratory system designed so that we can follow what's happening analytically in a very clear and simple way. But of course, the vesicles that form naturally aren't so nicely constrained. They're extremely heterogeneous, and that generates a system which is much harder to study experimentally. So that's illustrated by this image, which shows the range and the diversity of vesicles that are formed when you just shake up oleic acid with some salt and buffer. You get these huge vesicles, tiny vesicles, compound vesicles. It's a terrible mixture. And if you imagine adding some more oleic acid to this in the form of micelles so that everything grew a little bit, you couldn't tell that anything had happened. So we need new analytical ways of looking at this, or new ways of preparing vesicles so that we can actually watch what's happening. And our major advance in that area came from the work of a new graduate student, Ting Zhu, who had a background in mechanical engineering and proposed a very simple and elegant way of preparing more or less uniform populations of vesicles that were large enough to just look at in the microscope. So for the first time, we were able to do experiments where we take vesicles, add food, and just watch how they grow. And so what you can see in this image is a field of vesicles. They're all roughly 4 microns in diameter, so big enough to image optically. And what you're actually seeing here is a fluorescent dye encapsulated in the aqueous internal space of each vesicle. So now what you'll see next is a time-lapse video of how these things actually grow following the addition of more fatty acids. What we expected was simply that these spherical vesicles would swell up a little bit. We just watched them get bigger. We thought that if the surface area grew faster than the volume, they might become somewhat elongated in shape. But we did not expect what actually happened, and which is quite dramatic. And so what you see here is initially a faint filament emerging from the parental spherical vesicle. And over time, that filament grows and absorbs all of the material in the starting spherical vesicle, which has now grown into a long, thin, branched filamentous morphology. So this was a very surprising observation. It immediately raised a host of mechanistic questions, which we're still investigating. But it, very importantly, immediately solved the problem of how cell division might happen in a manner driven by environmental fluctuations. As I said, spherical vesicles are hard to divide. It takes a lot of energy. But these structures are incredibly fragile. And all you have to do is shake them up. Gentle agitation is sufficient to cause them to divide. And you can see that process in action in the next video. So here you see the purling instability. The filament forms a set of beads, and then they snap apart and generate fragments which eventually round up into spherical daughter vesicles. And so this process is driven by very gentle perturbations in the fluid, very mild shear forces. You can easily imagine the shear forces generated by wave action on a shallow pond leading to cell division in this kind of process. Now, this process of growth followed by shear-induced division makes a cycle that can be iterated indefinitely. So you can start off with a spherical vesicle, grow it into a filament, divide it into spherical daughter vesicles, which can grow again. If they sit around, the volume increases, they round up, then you can grow again, divide, grow. This cycle of growth and division can be carried on indefinitely. Okay, so let's think for a minute about some of the mechanistic processes that are involved. We don't understand everything that's going on, but at least we have some experiments that tell us what some of the critical factors are. Okay, so what we think is going on and what the important factors are is that surface growth is happening much faster than volume growth. And so that makes a shape change inevitable. Volume is conserved on the time scale of growth because most of the solvents are only very slowly permeable across these membranes. And the solvent can be anything from the rather artificial buffer that we use to a mixture of amino acids, such as you would get from Miller-Urey type syntheses. The other and somewhat surprising factor that's critical is the multilamellar structure of these vesicles. So these large vesicles tend to have several bilayers closely opposed as their sort of outer structure. What we think happens is that when we add food molecules, they first integrate into the outermost bilayer. There's very little space between it and the next bilayer. And so that extra surface area is extruded as a thin initial filament, which then subsequently elongates into this long tubular filamentous structure. We really don't understand the physical forces that are involved in those later stages, but we have some ideas about the initiation phases. So I want to show you first an experiment that shows that volume conservation is very important. If you use a buffer that is very, very rapidly permeable, then you don't see this kind of process at all. Instead, what happens is that the outermost bilayer just balloons up. As surface area increases, the volume keeps up, the volume expands, and so you end up with most of the bilayers almost unchanged and trapped within a much larger unilamellar vesicle. Now, the second important aspect is how multilamellar vesicles grow in contrast to how unilamellar vesicles grow. So when you have these multiple bilayers, as I suggested, it looks like from these confocal images that it's the outermost bilayer that grows first, and that extra surface area is extruded as a thin tubule. Eventually, you get a long filament, and when you divide that by mild shear forces, you get back small multilamellar vesicles, and then you can repeat the cycle. Now, something very different happens if you go to the trouble of making a unilamellar initial vesicle. This grows the way we intuitively thought it would grow. It just expands, becomes somewhat elongated. But now these structures are extremely delicate and unstable. And when they're subjected to shear force fluctuations, they tend to rip apart, the contents spill out, and remarkably, they reassemble into multilamellar structures. So to summarize this, the two important aspects of the overall process are beginning with multilamellar vesicles and having something in the surrounding solution which can only permeate slowly across the membrane, resulting in slow volume increase compared to rapid surface area increase. And then environmental perturbations lead to cell division in a very simple and natural way. Okay, so to summarize this, in a series of stages, we've gone from a very artificial laboratory simulation to a process that looks simple and robust enough to have occurred on early Earth environments. Okay. Now, more recently, we've come back to the idea of competition between protocells as an alternative mechanism of driving growth. And this is particularly interesting in terms of a way that this could emerge from Darwinian processes. We think that one of the first ribozymes to emerge, perhaps the first, was actually a ribozyme that could catalyze the synthesis of phospholipids. Okay, and so why do we think this? This came out of considering the series of evolutionary transitions that must have occurred to get from primitive membranes based on fatty acids, which with the appropriate dynamic and exchange processes for a primitive cell, up to modern membranes, which are based on phospholipids, which are good barriers and which rely on highly evolved biochemical machinery to mediate growth and division and the transport of molecules across the membrane. So you couldn't go directly from the primitive state to the modern state. That would be suicidal because now a cell that depended on the influx of nutrients from the environment would no longer have access to those nutrients because these membranes are so impermeable. So presumably the transition had to occur in a series of stages, and what would an intermediate stage look like? Probably it would look like a fatty acid membrane with a small amount of phospholipid doped into the structure. So if you think of a protocell containing genetic molecules, if one of those sequences happens to be a simple acyl transfer ribozyme that could make phospholipids. After all, it's a simple one-step acyl transfer reaction. You could imagine making just a little bit of phospholipid as an initial step in this transformation. The thing that puzzled us for a long time is what possible selective advantage could there be to the presence of a small amount of phospholipid in a fatty acid background. It was very hard to imagine any reason why that would be advantageous. It didn't really seem that a little bit of phospholipid would change the physical properties of the membrane in any particularly useful way. And so the answer to this conundrum came from doing experiments, and this is all work done by Itai Buden, who's a graduate student in the lab. So what Itai did was simply to prepare vesicles containing a little bit of phospholipids doped into a fatty acid membrane structure. And then he asked, well, how would that affect the properties of the membrane? And what turned out to be particularly interesting is what happens when these vesicles are mixed with pure fatty acid vesicles. And it turns out, as you can see on the next slide, that even a little bit of phospholipid confers a really dramatic ability on that vesicle to absorb fatty acids from its neighboring fatty acid vesicles. So here's the experiment that Itai did. He prepared vesicles with 10% of a phospholipid in an oleic acid background and mixed them with a large excess of pure fatty acid, oleic acid vesicles. So the vesicle starts out as a spherical structure. You can see the encapsulated dye. Within seconds, you see the protrusion of small filaments. And over time, you can see a transformation into a filamentous structure, much as you saw before following the addition of a large excess of fatty acids. But this time, the process of growth is being driven purely by the initial vesicle absorbing fatty acids from its neighboring molecules. It's essentially growing by eating its neighbors. The mechanism of this phospholipid-driven growth is extremely interesting, and you can see what we think is going on in the next slide. It turns out there are actually two processes. The first is a simple entropically favored process in which the absorption of fatty acid molecules into the mixed bilayer dilutes the phospholipids, which are extremely insoluble in the surrounding space. So this is an entropically driven dilution. But it turns out that in addition to that factor, there's actually a quantitatively more important factor, which is the fact that these phospholipid molecules change the properties of the bilayer such that the dissociation of fatty acids from the bilayer is slowed down. As a result, the vesicles grow because the association rate constant is the same, but the dissociation is slowed down. So they hold on to more of their molecules, they grow, and the pure fatty acid vesicles therefore shrink. Okay, so how do we demonstrate this experimentally? Itai took advantage of an assay that was developed by Jim Hamilton. It's a rather indirect but very effective assay. So if you have a fatty acid vesicle, you can monitor the dissociation of fatty acid molecules from that membrane, which is a slow step, by looking at how they dissociate into solution, integrate into the membrane of a reporter vesicle, which is a fatty acid vesicle that contains inside it a fluorescent dye that's pH sensitive. Now when the fatty acid molecules integrate into the outer leaflet, they flip rapidly to the inner leaflet. It's the protonated form that flips because it's neutral. It can flip very rapidly. And then it re-equilibrates on the inside, releasing protons which acidify the interior. So we have a fluorescence assay for this pH decrease, and that gives us essentially a real-time assay for this initial slow step of dissociation. You can See that this assay works by comparing the dissociation of fatty acids of different chain lengths. And so here you see that oleic acid, which is 18 carbon chain, dissociates fairly slowly. Palmitoleic acid, 2 carbons shorter, dissociates more rapidly. And myristoleic acid, 14 carbons, dissociates extremely rapidly. So it gives you a good readout for the dissociation kinetics. And that allows you to monitor the rate of dissociation as a function of the phospholipid content of the membrane. And so if you start off with a pure fatty acid membrane, so a pure oleic acid membrane, dissociation is quite rapid. But the more phospholipid that there is in the membrane, the slower the off rate of the fatty acid is. Okay. So since as a result of these two processes, this entropically driven dilution aspect and this off rate driven aspect, the presence of even a little bit of phospholipid will drive growth by essentially extracting or absorbing fatty acid molecules from neighboring vesicles that have either no fatty acid or even less, just less fatty acid. So what would the implications of that process be in an evolutionary scenario? Okay, so we begin with this idea that phospholipids drive growth. As a result, of course, there would be a very strong selection for a ribozyme that has the catalytic ability to make phospholipids, so a simple acyltransferase ribozyme. That kind of primitive cell would rapidly take over the population because it has such a strong selective advantage. But that would lead to an evolutionary arms race, because now, in order to grow by eating your neighbors, you have to make more phospholipids than they do. Okay, so there would be a selective, an evolutionary race to make more and more and more phospholipids. And so now the phospholipid abundance in the membrane would start to increase. You'd have more and more phospholipids. And eventually that would lead to new selective pressures favoring the emergence of metabolism, because now it's actually useful to have internal metabolic reactions because you get to keep the molecules you make instead of having them just leak out, since the phospholipid makes the membrane less permeable. At the same time, there would be selective pressures for the emergence of transport machinery, which would help you absorb molecules that you still want to have access to from the environment, or release molecules that you want to get rid of, so waste products. So we think that this physical process, this unexpected physical process of phospholipid-driven membrane growth would set in place a whole cascade of events, eventually leading to selective forces for much more complicated cells that, where we see the emergence of metabolism and membrane transport machinery. And eventually, we think that could drive the emergence of protein synthesis, initially uncoded but eventually coded protein synthesis, and be a major factor in the emergence of modern biochemistry. Okay, so from all of these experiments, just to sum up, what can we say that we've learned that's really relevant to the origin of life on Earth? And I think at a general level, there are two things I'd like to point out. One is that from doing very, very simple laboratory experiments, again and again, we've uncovered a surprising new physical phenomenon, things that you would have a hard time thinking up a priori, but which emerge naturally from doing laboratory experiments where you're trying to build systems and see how they work. The second general lesson, I think, is that in our early experiments, we tend to use very highly constrained artificial systems because they're analytically tractable. But they often don't exhibit the properties that we really want to see that are relevant to the prebiotic system. So it's very important to relax those experimental constraints and gradually develop the ability to look at messier and more natural systems that give you insight into physical processes that really could explain some of the apparently hard parts in the process by which life emerged from the chemistry of the early planet. Okay. So again, all of this work was done by many very talented students and postdocs. I've mentioned in particular the work of Itai Buden and Ting Zhu, and the early work of Irene Chen, Shelley Fujikawa, Marty Hanchek. Many people have played a role in developing these systems and gradually giving us the ability to understand what's going on in these model systems and some insight into the origin of life. Thank you.